Assistant Lecturer of Radiodiagnosis, Alexandria University. She's going to talk about molecular MRI imaging in neuroradiology.
Our topic today is about molecular MRI imaging in neuroradiology. First of all, conventional MRI imaging relies mostly on the anatomical, physiological, or the metabolic properties that distinguish between normal and pathological tissue on a macroscopic scale, whereas molecular MRI imaging, on the other hand, is focused on the direct visualization of specific population of cells or molecules on account of their metabolic processes occurring at a submicroscopic level. The purpose of molecular imaging is not to define the anatomical features, but rather the detection of the metabolic events that are responsible for the early progression of the disease. Different types of functional and molecular MRI imaging methods represented as the PET MRI, the hyperpolarized methods, dynamic contrast enhancing, targeted agents, diffusion-weighted image, magnetization transfer, cell labeling, and MRI spectroscopy. The first topic we will talk about is targeted contrast agents. MRI contrast agents are molecules or particles that influence the relaxation of the water protons. They can be either T1 positive or T2 negative weighted, affecting the longitudinal or the transverse relaxation times respectively. T1 weighted contrast agents are either gadolinium or manganese paramagnetic complexes, whereas the majority of the T2 weighted are iron oxide based supramagnetic nanoparticles. Due to the lack of sensitivity of MRI, relatively high concentration of contrast agents is needed to be injected to generate local variations in the water signal intensity and therefore a detectable signal. However, targeted contrast agents will cause the accumulation of large number of gadolinium at the site of interest by using macromolecular system, example polymers or nanoparticles, and the use of target probes which their efficiency changes when binding to their biomarker. As we said before, the T1 weighted contrast agents will lead to enhancement in the lesion, whereas the T2 weighted MRI agents will lead to the suppression of the signal in the lesion. Different targeting moieties can be used to achieve a specific binding to a chosen biomarker. For example, in case of tumor imaging, they may bind to cell surface receptors that are specifically expressed on the tumor cells or the receptors found on the endothelium of the tumor vessels. This is the gadolinium particle. We will couple it with a small molecule, antibody, protein, or peptide, and it will bind to a specific biomarker. And the example of these biomarkers include the folate receptor that are overexpressed in the ependymomas and medulloblastomas, and other agents will bind to the transferrin receptor by conjugating iron oxide nanoparticles to the iron transport protein, and can be used for detection of nerve sheath tumors or brain tumors which overexpress the transferrin receptor. This image is of a post-folate receptor targeted iron oxide. It shows marked suppression in the signal of an adenocarcinoma that was present in a rat model in the soft tissue. Other uses of targeted contrast agents, specifically the supramagnetic iron oxide nanoparticles, is the image labeled cells using MRIs. The cells will be labeled by incubating them in a contrast medium outside the body, or an alternative approach is to have label taken by phagocytes following the intravenous injection of contrast medium. Oligodendrocyte precursor cells is an example of this, which has a high promise as a transplant population to promote the regeneration of the central nervous system, specifically to produce myelin in cases of multiple sclerosis and spinal cord injury. This is the image of the oligodendrocyte precursor cell, which at the end will become a myelinating oligodendrocyte. The second topic is the chemical exchange saturation transfer. These agents represent a new class of MRI probes which can be switched on and off on demand. Following injection, a selective low-power radiofrequency pulse is applied to the cyst agent which saturates its signal. Given that this agent is likely to be of a low concentration, the signal intensity will be difficult to detect in a biological system, and therefore an alternative method is required to visualize it. The proteins in the surrounding water are easily detectable, and since they are exchanging with a cyst agent, the saturation will be transferred into the bulk water, producing a visible reduction in the signal intensity. Therefore, changes in the intensity of the water signal is indirectly reporting the molecule of interest. Depending on which mobile proteins are used to generate the signal, several techniques are possible, and the most common being the amide cyst, or also what is known as the amide proton transfer. As we mentioned, this is the molecule of interest with an amide group. After the application of a radio frequency pulse, there will be a saturated proton, and this proton will be exchanged with the surrounding water molecules until they will all saturate, and this will lead to a measurable reduction in the water signal intensity. These are examples of the amide proton transfer maps. This is a case of a medulloblastoma. It shows very high intake in the amide protein transfer map. While this is a low-grade glioma, which shows no post-contrast enhancement in the T1 image, yet detectable changes were present in the amide proton transfer map. Another case of an IDH wild-type diffuse astrocytoma showing no post-contrast enhancement in the T1 weighted image, yet changes in the map of the amide proton transfer was also detected. The last technique we'll talk about is the hyperpolarization technique. The major disadvantage of MRI is its low sensitivity when compared to techniques such as the PET. When protons are placed into a magnetic field, they enter one of the two energy levels. Approximately half will enter the lower energy level, and the other half will enter a higher energy level. And the small difference between these two poles is what is used to produce the MRI imaging. If some of the nuclei that are not being used to produce the imaging could be made use of, then the sensitivity of the MRI will be increased. And if all the available nuclei could be utilized, the sensitivity of the detection will be multiplied by a factor of 10 to the power 5. And this is what is known as hyperpolarization. The commonly used metabolite for hyperpolarization is carbon-13. It's an emerging clinical technique with the potential to increase the understanding of neurological, psychiatric, and neuro-oncological condition by probing the cerebral metabolism. Carbon-13 pyruvate, which informs upon the oxidative and the glycolytic metabolism, is the most commonly used. In the Krebs cycle, the metabolism in the mitochondria is an efficient process for ATP generation, while the glycolytic metabolism will result in the formation of the lactate by the lactate dehydrogenase. Here is the carbon-13 pyruvate. It will take two pathways. Either it will be converted into lactate, and this occurs in the glycolytic metabolism, which commonly occurs in cancers, or it will enter the Krebs cycle in the mitochondria, and the end result will be bicarbonate. The process of hyperpolarization involves the mixing of carbon-13 labeled metabolite substrate of interest to a source of free radicals, a source of free electrons, which is known as a free radical. The sample is then stored inside a sterile unit known as a fluid bath and placed inside a magnetic field in a bath of liquid helium while undergoing irradiation with a microwave source. These conditions will increase the available signal from the molecule in order of 10,000 folds. This is the machine where hyperpolarization occurs, and as we said, the carbon-13 pyruvate will be subjected to microwave source, and multiple free electrons will be coupled to the carbon-13, augmenting its signal. This is an image of these are two patients presenting with glioblastoma multiforme post-management, one with a progression of the tumor and one with a stable disease. First of all, the T1 post-gadolinium in the first patient shows marked heterogeneous enhancement. Here, it shows less enhancement. The lactate pyruvate map, it is here markedly increased than this stable disease. As we said, the lactate is very common to increase in cases of cancers, whereas the bicarbonate pyruvate didn't change in these two patients. Thank you so much.